Hi, and welcome to VWR. Your search is over. Whether you're a university researcher, an industrial medicinal chemist, or an independent innovator pursuing your dream at a startup, VWR is proud to bring you all you need for organic synthesis. From state-of-the-art fume hoods to specialty glassware, to building blocks, catalysts, and dry solvents, to flash, prep, and thin layer chromatography products, as well as relevant equipment and instruments and lab supplies. You will find unparalleled product choice at VWR. In addition, VWR offers a complete range of safety, personal protection, and waste disposal products, as well as technical, validation, and e-business services that can help you further optimize your logistics and purchasing functions. Product choice, differentiated services, and operational excellence is what makes VWR the one supplier that delivers exactly what you need so you can focus on your work. One powerful tool designed to reduce the time and complexity involved in finding and ordering organic compounds is the VWR Building Blocks Portal, powered by eMolecules. VWR's product and service solutions, plus eMolecules' data and chemical sourcing expertise, combine to deliver a streamlined search, source, and order process for chemical building blocks, so you can focus on your discovery. And that's just one of the many novel solutions that VWR has created specifically for organic synthesis. Explore them all on VWR.com.
Hello, everyone. Thank you for joining us for CEN's second annual virtual symposium, Advances in Drug Discovery and Development. I'm Mitch Garcia, an assistant editor at CEN, and I'm delighted to introduce our next speaker, who is Christopher Austin from the National Center of Advancing Translational Sciences. Chris will be presenting a talk entitled Catalyzing Translational Innovation. I'll be back to moderate a Q&A session immediately following the presentation, and we welcome your questions at any time. Just click the green Q&A button in the left corner of your screen. And if you need any technical assistance during the presentation, please click on the support button located top right of your screen. Please note that CEN does not endorse any company, products, or services that may be mentioned in this presentation. And finally, this talk will be archived and available on demand at the conference website for 3 months after the live broadcast until December 16th, 2015. And now I'm delighted to hand over to Chris.
All right. Well, thank you to everybody for joining us. I'm Chris Austin. I'm the director of the National Center for Advancing Translational Sciences, and I'm going to run through with you in the next half an hour or so what NCATS is doing in the areas of not only chemical sciences but drug discovery and development. And I decided to make this purposely broad. Rather than talking to our chemist colleagues only, I decided to talk to the whole community. So I'm going to include some of the clinical work that we do as well, so you'll have a full sense of what NCATS does. So let's just jump in. Sometimes I'm asked, why NCATS? NCATS was started as the newest component of the NIH about 4 years ago, a little bit less than that. And among the reasons was this one that I think we're all painfully aware of, that is that we really live in a paradoxical time scientifically and medically, that on the one hand, we know more about ourselves and health and disease than we ever have. Perhaps exemplified by the Genome Project, but there are many other examples. But if we ask, well, have the epochal changes in science and medicine that have happened in the last 50 years been paralleled by epochal changes in health of the American population or any population? For the most part, the answer to that question is a resounding no. And to some degree, that's because we really don't understand very well how to go from what's on the left to what's on the right. And there's a lot of knock-on effects of this, but among them are that people are unhealthier than they should be, and funders, frankly, of the biomedical research enterprise have either gotten impatient or have lost patience with us completely in terms of our ability to deliver, at least on this part of our mission. The good part, however, the good side of what's happened in the last 30 years, really since I was in training, is this, which is a rather remarkable graph. This is looking at the number of disorders in the online inheritance in man database from NLM and CBI. And this is what the graph looks like. That is from the early to mid-'80s, when the number of disorders that molecular basis of which was known was a handful, going back to the original one, which was sickle hemoglobin by Linus Pauling in 1949, and then probably a lot of you remember cystic fibrosis in 1989, then Huntington's disease in 1990, 1991. This number really started to go up when large amounts of sequence data started getting put in the in the public databases as part of the Genome Project. And the remarkable thing now is 30 years later, there's almost 5,500 disorders of the 7,000 or so disorders that affect the human family, the molecular basis of which is known. That's the good news. The bad news is that there are treatments for only about 500 of them. Now, part of this is, of course, due to the fact that, as we all know, probably on this webinar, takes somewhere around 10 to 15 years after a target is identified and validated before a drug would be available. But actually the pace of diseases becoming treatable for the first time has actually not changed in the last 15 years. It's still about 3 to 4 a year of these diseases that go from untreatable to treatable. Most new drugs are approved for conditions that already have a drug approved for them. So when NCATS was formed to try to address this problem, we had to first define what the heck translation is. This is a sort of an unfortunate word because it has many meanings, particularly non-scientific meanings. A lot of you will be amused by the fact that when I became director of NCATS, I was asked whether the National Center for Advancing Translational Sciences studies linguistics, because of course to any normal person who's not a scientist, that's what translation means. So we spent some time defining what we mean by translation, and it's really a quite broad definition. It's the whole process by which an observation that's made by a scientist in the lab, a doc in the clinic, or a public health person in the community makes the whole process of turning that observation where a light bulb goes off and says, this observation might be able to, it gives me an insight that might be able to address this particular scientific problem or health problem in an effective way. The whole process of creating that intervention, that drug, that device, the behavioral intervention, and then showing that it's useful in the health of individuals and then in the public, that whole process from idea to public health is what we define as translation. But you notice that the NCATS is not the National Center for Translation. Every center at every institute, 27 institutes and centers at NIH for the most part do translation, and of course many organizations outside of NIH, of course the whole biopharma industry does this, and so we're not the National Center for Advancing Translation per se, we're the National Center for Advancing Translational Science. So what is translational science? Well, we define this as the field of investigation that seeks to understand what the general principles are by which this whole process happens, and both scientific principles and operational principles. I think all of us who work in this field are again painfully aware that this is it's a remarkably empirical process, mainly a remarkably trial and error process. We really don't understand the science underlying most steps of the translational process. And our hypothesis is that unless we can understand those general principles, translation will remain an empirical, phenomenological trial and error, and mainly error process. And only by understanding the general principles will we be able to turn it into a predictive science and therefore be more efficient, lower cost, faster, etc. So with that vision, here's the NCATS mission. I don't want to dwell on this, but I just want to emphasize a couple of words. The first is the word catalyze. This group knows what a catalyst is. Catalyst, of course, is an entity that brings together reactants that don't work by themselves and allows them to produce a product that they otherwise wouldn't by themselves. And that's really what NCATS does. Everything that we do is a collaboration with someone, either at NIH or in industry, a nonprofit, FDA, etc. And what are we catalytic of? Well, we're generation of new ways of doing things. It's very clear that that the biggest impact in science comes from new methods, new technologies, new ways of doing things, and that's really what NCATS aims to do. It's important that I mention this on the catalytic part, that everything we do is a collaboration, and there are in fact 27 institutes and centers that make up NIH, and they all have different names, mainly after organ systems or diseases that they work on. And the important thing is, from the very beginning, NCATS was viewed as, envisioned as, and is operated as. We operated as a collaborative instrument and a convener, if you will. But because of the position that we occupy in the research ecosystem, we have more regular and perhaps deeper collaborations with organizations outside the NIH than many of our colleagues here. And those are not only with our conventional colleagues in academia, but also with disease advocacy groups, nonprofits, other government agencies like the FDA and the EPA, pharma, biotech, VC, etc. So now that we hopefully understand the purpose of NCATS, that is to put translation on a solid scientific and operational footing, and thereby get more treatments to more patients more quickly, what are the problems that we work on? And this is a partial list of the problems that we work on. I want you to notice a couple things about this. One is that they're variously well known as being the causes of either drug development or intervention development failure, or implementation failure at one level or another, or implementation inefficiency. And the second thing is that there are no disease names in this list, and this is really the point. Everything NCATS works on are in what economists would call the tragedy of the commons. That is, these are problems that are everybody's problem in general, but nobody's problem in particular, so nobody's ever worked on, really, because it's been nobody's day job. These are our day job, and so they are things that we work on as our primary mission. But it's important that they're not just scientific problems, they're operational problems too, organizational problems. Translation literally means linguistically. It actually means to carry across, and so translation by definition means we are going from one organization or one interest group, if you will, to another, whether that's from biologists to chemists, or chemists to informatics people, or the preclinical team to a clinical team, or from clinical to regulatory, or regulatory to physician groups or practice groups, etc. There's always a handoff going on, and so whenever that happens, it makes the organizational challenges just that much greater. And we view these as areas of scientific and organizational study as well. And these are just some of the things that we work on that are limiting to translational efficiency in that domain. So in the scientific domain, so now that we have sort of the rationale and what are the problems that we work on, how do we actually get things done? It will probably not surprise you that we've divided the activities broadly into clinical activities. Those are in human subjects, human participants. In the preclinical space, that is going from target validation into really first in human studies, and then work to re-engineer the whole translational process. And I'm just going to show you an example of each of these to just illustrate the nature of the work that we do. First, on the clinical side, and I'm going to spend the least time on this because I think it may be of a less familiarity, perhaps, or less relevance to this particular audience, it's very clear that even if we had, just as an example, a drug for every untreatable disease, those 6,000 or so untreatable human diseases, if we had a drug for each one of those diseases tomorrow, we in no way have a system in this country to be able to test whether those drugs or any other intervention is safe and effective. And there's a lot of reasons for this. Some of it is because of our atomized healthcare system. But the important thing is that the research that needs to be done to translate a novel intervention into human studies of safety and efficacy, and then into populations, is underdeveloped at best in this country. So there's a program called the Clinical and Translational Science Award Program. It's actually the NIH's biggest single programs within NCATS, which has within it 62 academic medical centers that we call hubs. Many of the large, well-known academic health centers are in this program. So it's an unprecedented collection of key opinion leaders and close to 100 million patients cared for across this network. And what we put in place more recently is innovation centers that address some of the real limiting reagents to being able to get trials done. Those include things like multi-site central IRB human subjects review, contracting, budgeting issues, which can hold up a trial getting started for up to 2 years. If you can imagine, if you're a basic scientist, imagine that you come up with an idea, but it takes 2 years to get through all the approvals that you can actually start your study. Science, fundamental science, preclinical science would advance at a snail's pace, and this is really the state of the art in science, in the clinical world. In the green circle there illustrates our solution to one of the other problems, is that even when one can get all the approvals, upwards of half of clinical studies which are started are never completed because enough participants are not ever recruited to actually answer the experimental question that was asked in the first place. So a remarkable waste of time, spun wheels, patients exposed to potentially harmful or helpful interventions without any information coming out of them about whether they were actually effective. So again, tremendous waste of time and effort. So the whole idea is that via this network, over the next couple of years, we will have put together a true national network for translational medicine that will allow us to allow the research ecosystem, public sector, private sector, nonprofit sector, to be able to come to this network and, like the picture on the lower left illustrates, be able to treat the CTSA program, that network, like a supercomputer into which a sponsor could plug their USB and get a trial started in kind of a just-in-time way. So that's one of the large innovative programs that NCATS is working on. Works on sort of the lower half of that problem list that I showed you a minute ago. Another clinical program that we have, and I'm going to move backwards into the preclinical space. Another clinical program is focused on rare diseases. NCATS is interested in rare diseases for several reasons. One, they are representative of represent disproportionate unmet medical need. I mentioned that there are about 7,000 human diseases. About 6,000 of those are rare, and all but about 250 of them have no treatment. So the question is how to accelerate the rate of discovery and development of interventions which can treat these very numerous rare diseases. It may interest you to know that even though rare diseases are individually rare by definition, less than 200,000 by the FDA definition, given the fact that there are about 6,000 of these disorders, the total number of patients in the U.S. that has a rare disease is somewhat north of 10 million people. So it's a significant issue from a public health standpoint. So this rare disease clinical research network has just two characteristics that I want to illustrate for you. One is these little ovals called PAGs. This means Patient Advocacy Group. This has been a really critical innovation for this network. That is that you can't get funded in this network without having a patient group as a research partner. That has been invaluable at speeding up and focusing the work of these programs, and also bringing urgency to the problems that are being addressed. The other is that you will notice that if you look at all the different names, and there's 22 different consortia, you'll notice that they don't work on individual diseases. And this is on purpose. These 6,000 rare diseases are, of course, don't exist in isolation. They're all related to each other in one way or another. So all of these programs were asked to organize rare diseases, some corner of the rare disease space, by some organizing biological principle, either a biochemical pathway, for instance, the urea cycle disorders there on the right, or an organelle, cellular organelle, like the mitochondrial disorders consortium or the lysosomal disease consortium, or a phenotype like a rare kidney stone or vasculitis. And this program has, I think, because of those two things, because of the involvement of patient groups and the involvement of the organizational principle that we're talking about, has really been remarkably successful. Moving into the preclinical or bordering clinical preclinical space, some program that you may have heard about is this new therapeutic uses program. The idea here is that about 8 out of 10 drugs that are developed in pharma or biotech that go into people are never approved for one reason or another. So the idea was, could we partner with these pharmaceutical companies and hook them up with academic investigators that might have novel ideas, novel indications to which these investigational drugs could be put, and then test them in a rapid fashion. And so this is a three-way collaboration between pharma, academic researchers, and NCATS. These are the diseases that were the projects that were funded about a year and a half ago now. You notice that they are on mainly untreatable or poorly treatable disorders, and they have an academic partner and a pharma partner. I just want to illustrate one example here. It's a really interesting story that we think may be an exemplar of things to come. This is story about an AstraZeneca drug, a Src kinase inhibitor that was initially developed, of course, for cancer. Didn't have great efficacy as a standalone agent in cancer, so it was deprioritized by AstraZeneca. An investigator at Yale, about the same time, discovered that a Src kinase family member named Finn seemed to be involved in Alzheimer's pathology. And so because of this program, was able to go from that discovery of Finn kinase and Alzheimer's disease to testing the hypothesis in animals and now in humans about Finn inhibition ameliorating Alzheimer's pathology in about a year and a half, which is, of course, significantly shorter than would have taken to develop an NCE from scratch. Moving further back now into the division of preclinical innovation, which is, I think, where a lot of us in the ACS operate on a daily basis. We have a collaborative program which has investigators from around the world who are stuck at these project entry points at the top, these arrows, and they get stuck at various places for various reasons, either expertise or experience or facilities. And they come into one of more of these programs in the little boxes there, and they partner with NCATS scientists to move the project forward, generate data or reagents or drugs, investigational drugs, to move these projects forward. And I'll just give you a couple of examples. This was one early effort in repurposing. We're always trying to develop general principles, of course, which are going to improve the efficiency of the translational pipeline. It was very clear. That, of course, virtually all drugs have off-target activities or other activities from that which they were originally intended some good, some bad. And the good side, that of course can lead to repurposing, but this is has been a mainly serendipitous exercise. So we thought, well, gosh, what we ought to do is put together a complete non-redundant list of every drug ever approved for human use worldwide. This had never been done about 6-7 years ago when we started this. This is now done. There's a website on our that you can get to. There's all the database is on our website. You can download the whole thing. This is a paper from a couple of years ago that describes this. And I'll just give you one example of where this has been put to. This story from a couple of months ago. This is a collaboration with a liver researcher, happens to be at the National Institute for Diabetes and Digestive and Kidney Disorders on hepatitis C. You're all aware, I think, of that disorder and the new drugs which have come out treating HCV. Very effective, but of course quite expensive. This initiative identified this old first-generation allergy drug, chlorcyclazine, as being a very effective inhibitor of hepatitis C replication in cells, and does not hit any of the targets that have been identified before. Now that's clear. We're still working on what the target is, but the important thing is that this is already an approved drug, might be complementary to other therapies, and of course would be quite cheap, given that it's been around for about the last 50 years. We do a lot of patient-driven science. A lot of the uses of the drug collection that I talked about come not from scientists, although we work with hundreds of academic and nonprofit and for-profit researchers. But they come from patients too. This one on Charcot-Marie Tooth, this one on chordomas, a rare cancer, and this one on a muscle disease called myotonic dystrophy actually started because patients who had these disorders came to NCATS and partnered with us to get these projects going. And this has been a remarkably effective counterpart to the more conventional academic or scientifically initiated collaborations. I just want to give you one example of a novel chemical probe development, because this is something we do a lot of as well. We work a lot on the approximately 90% of the genome which is undrugged, moving targets from the undruggable to the druggable category. We really don't work on things that already is being worked on, because there's most of genome and disease space is not worked on. So this happens to be just an example of several, actually literally 100 that I could show you. This happens to be on inhibitors of an enzyme called 12-lipoxygenase. It's on the arachidonic acid pathway. It was identified about 30 years ago, but because there were no selective chemical inhibitors, it really wasn't understood what this enzyme did. And so this part of NCATS called the NCGC collaborated with an expert in lipoxygenase at UC Santa Cruz. Over the course of about 2 years, developed an assay, did a high-throughput screen of about 400,000 compounds, did the chemical informatics, and about 2 years of medchem, leading to these enantiomer-specific, first enantiomer-specific 12-HLO inhibitors, and using those probes, and then we were able to dissect what this enzyme actually does in cells and then in tissues. Turns out it's involved in both diabetes and thrombosis. That then led to collaborations with people in those fields, and then a research collaboration agreement with Sanofi to further characterize and develop those inhibitors. So there's a great example of a target that we started working on simply because it was undrugged, and we're able to dissect novel biology and potentially new therapeutics as a result of it. And lots of papers, of course. This one happens to be from J. Medchem. Let's say, however, you are at that point where you develop a probe or a repurposed drug, but it's not for a common disease like thrombosis, but for a rare disease. There rarely are going to be companies that are interested in those probes. So this is a program to go from a probe to first-in-human development again, and it's this collaborative way that NCATS does everything. Here's just one example of that. This is a compound up there in the upper right corner, was initially identified at Virginia Commonwealth University about 20 years ago by a researcher who's now retired. It was not developed by that researcher. The IP was acquired by this little company in Boston called SRX. It's a kind of a scary compound from a chemical standpoint. It has also a potential very interesting but potentially problematic mechanism, which is shown in the graph below, which is that it binds to sickle hemoglobin and shifts the oxygen dissociation curve to the left. What that means is that it reverses the fundamental molecular defect of sickle cell disease, which is very attractive. But the question was, would this compound bind to many other proteins in the proteome and cause toxicity? It didn't, but that was the concern. And so this compound in this company was dead in the water, not able to move it forward, even though sickle cell is a very important and from a mechanism standpoint is untreatable disorder. TRND, this part of this Therapeutics for Rare and Neglected Diseases program, part of NCATS, partnered with this company, did all the tox CMC regulatory work, got into humans in about a year. From signing the agreement, got through the phase 1 and phase 2 trials, and as a result of this, the whole program was acquired by Baxter for completion of clinical development once it had been de-risked initially in humans. I'm going to skip through this tox part quickly. I just want to let you know what's going on here. There's a large tox collaboration that we're doing with the National Toxicology Program, the EPA, and the FDA called Tox21. It's a massive systems biology experiment testing about 10,000 chemicals, drugs and environmental chemicals for the pathways that they interact with as a way to understand the toxicity of chemicals and eventually to develop predictive models for biological response in humans. Important things for all of you on the phone call is that this is a community resource project, and all of the data from this program are put in public databases, either ACTR from EPA or PubChem from the NIH. There have been about 100 million data points from this program. That is a chemical at a concentration against a biological assay that has been made public in the last 3 or 4 years from this program. So if you're a modeler, please go to it and tell if you can come up with novel predictive models based on all these data. I just want to finish with this, which is gives you a sense of where NCATS is going. This is one of the re-engineering programs. Another approach to the toxicity problem that's complementary to the Tox21 program is this program to develop three-dimensional organoids of all human tissues that are targets of xenobiotic toxicity. It's a program which is developing microfluidic on a microfluidic platform models of all of these 10 organs that you can see there in front of you, and they have to be anatomically and functionally correct and work for at least a month. To give you a sense of how this works, they all start with an extracellular matrix of some sort. The cells are added. They're either primary cells or iPS cell-derived cells. They have to have a given structure that's either given to them or they self-assemble depending on the organ. Of course, they have to have the right spatial and temporal patterning. Sometimes they have to be perfused because, remember, these are alive for a month. They have to be in the right bioreactor to keep them alive during that time. They sometimes have to be innervated in order to give either the efficacy or toxicity readout that we're interested in. They frequently have to have a host response, especially if we're interested in toxicity. They have to have a particular functional readout that's non-destructive, and we have to have a computational design that allows us to gather the information. And all of this has to be done in a modular way so that we can mix and match these various organs. And I'll just give you one quick example. This happens to be the heart. In this example, cardiomyocytes, you're probably well aware, differentiate from iPS cells quite readily. However, they don't mature into mature cardiomyocytes. They remain rather immature, and therefore their responses to drugs are not terribly consistent with what happens in vivo. However, if one takes these a sheet of these cells out and exposes them to the same stretch and relaxation that a normal heart would undergo, actually this is how these cells actually differentiate, form sarcomeres, form Z-bands, and actually begin to respond to drugs in a more physiologically relevant way. So where we're going here is in the end of the next 5-year funding period, and we're beginning to do this already, to have all 10 tissues on a single microfluidic platform where one could, for example, infuse a drug meant there in the little blue circles there in the bottom right of the figure in artificial blood would go to an artificial gut and get absorbed or not, go to an artificial liver and get metabolized or not, and then go to an artificial kidney tubule, for instance, and cause tubular necrosis or not. Just as an example, one can put in or take out drug at any phase of this to do analysis, for instance, do analyte analysis, etc. And of course, one doesn't need all 10 together. One can put heart and lung together, or liver and kidney together, whatever one would want. And of course, all these readouts have to be correlated with the in vivo readouts, which is what's going on now. There's a lot of work going on to test whether these systems respond to both approved drugs and drugs that have failed in ways that mirror the clinical response in humans. The other interesting thing to sort of leave with you is that because each of these chips is made from either primary cells or more commonly iPS-induced pluripotent stem cells, these chips can be made from individuals. That is, each of us on this webinar could have their own chip made, and therefore one can begin to look at inter-individual variability in ways that are simply impossible currently. So I think that is it. I'm going to try to go, I went backwards unfortunately, so I'm going to go forward here. I think that was, yeah. So that is it. I'm glad to take any questions if we have time. If there's, of course, if you're looking for more information, we have a good website. We just redesigned a lot of information, contacts, all the people that you might want to meet there, how to work with us, etc. And at that point, I guess I'll hand it back over to Mitch, and we can take any questions.
Hi, Chris. Thank you. That was a great and very interesting talk. We have the first question is from Roberta Brinton from University of Southern California. They were wondering, what is NCATS doing to help change academic, publishing, and funding incentives to promote collaborative, transparent, and reproducible research?
Day meeting we're having here today about that very issue. A lot of this is going on through the CTSA program, which is resident at most big academic medical centers. And yes, the goal is both initially to have these centers appreciate some fields, some disciplines which are non-traditional, among which are medicinal chemistry, for instance, at many places, entrepreneurship, regulatory science, these kinds of things. But also to help them realize that translation, as we like to say around here, is an obligatorily team sport. And in a world that is still, for the most part, in many places at least, run on whether an investigator has a first or last author publication in a one-word journal, and those are required for promotion and tenure. Really, translation is going to be limited in its efficacy in those environments, because the middle authors, those really critical people in any team, are not going to get academic credit. So we're working very hard on that. Our long-term goal is to have a robust academic discipline of translational medicine, which has probably different culture, different promotion and tenure requirements, because the science that it does is different. It's not better or worse than fundamental science, it's just different.
Okay, our next question is from Vasu Devan from the National Chemical Laboratory. And they're wondering, in case of repurposing of a drug, how can one deal with IP or intellectual property issues?
It's interesting. It's a question that I get asked a lot. I would say there's a couple of different answers. First of all, the collection that we have, because it represents the historical accretion of every drug ever made, is made up mainly of generic drugs. About 90% of the collection is off patent for temporal reasons. Compounds that are on patent, of course, we need to and actually want to go to the originator company, the innovator company, and work with them to develop the compound. And that's what the new therapeutic uses program does. The cicatinib program that I mentioned, that's the sarc kinase family inhibitor that's being repurposed for Alzheimer's disease. That is a very close partnership with AstraZeneca and Yale working together. On the more common problem we have, actually, is that there is no intellectual property on these generic drugs, and that makes it very difficult to fund the clinical trials, the definitive clinical trials which are required for registration. That is a problem that we are still working on, and I don't pretend to have an answer to that. There's no way to recoup the investment required, at least 10, closer to $100 million sometimes to do a registration trial, depending on the disease and the drug. There's no way one can do that in a return on investment environment. It's a too large amount of money for NIH Institute or Center to do, really, for the most part. And it's too much money for a nonprofit, too. So we've been talking with many different partners about different kinds of incentives, different kinds of work that we might do to look at evidentiary standards rather than regulatory approval per se, to get these into medical use.
So we received two very similar questions. I'm going to do my best to kind of digest it. Both are from Oliver Grunman from University of Florida, and it's from Seng Shi from Chatham University. And they were wondering what type of training or education would be suitable for future researchers that want to be more involved in translational research.
Great question, and thank you for the question. It's something near and dear to my heart and everybody here. First, I should say that it will not surprise you to learn that I think this is a wonderful time for investigators who, especially younger investigators who are interested in getting into this space. And I know probably all of you are tired of hearing this, but it really is true that the opportunities available now compared to when I was in training 30 years ago are just mind-blowingly larger, and I tried to illustrate that at the beginning. I would say a couple things. First, it's important to have some expertise that's deep expertise that you can bring to the team, whether that's in biology or chemistry or pharmacology or informatics or clinical studies or whatever, public health, what have you. But you also need to learn, unlike in at least the way I was trained, you need to learn enough about all of those other areas that I just went through to understand what your teammates are going to do. And so the way I sometimes explain this is it's like learning how to play on an American football team. That is, you can be the best quarterback in the world, but you really have to understand what the center does, what the offensive lineman does, what the wide receivers do, or else you're never going to score any touchdowns. And that's a bit different from how scientific training has typically been done, which is in a very purposely narrow way, in a narrow field. That is still appropriate for folks who are going to be looking at fundamental mechanisms, where that training is often fine. But for translationalists, it simply doesn't work. The other thing that you should think about is a way to either learn about, or even better, spend some time in places outside of academia that are part of the translational ecosystem. And by that I'm including pharma, biotech, venture capital on one side, regulatory agencies like the FDA on another, patient groups who are increasingly both driving and funding some of this work in very sophisticated ways. It's really important to understand their points of view in order to be both psychologically and culturally multilingual, as well as scientifically understand where they're coming from. So it's I guess the way another analogy that might help is, you need to think about yourself the way maybe you did when you were in college. That is, that you had a major which you knew a lot about, but you also had perhaps distribution requirements, and you didn't know a little bit about everything, about math and science and humanities and art and all these other things. So you had a broad background on all fields, but then you had a major that you knew specifically something about that allows you to be a good team player.
So that's all the time we have. Many thanks, Chris, for your presentation, and thanks to all of you for tuning in. There'll be more great talks beginning at the top of the hour and throughout the day. You can find a full program on the agenda page, located at the upper right-hand side of your screen. In the meantime, please stop by the virtual exhibit and the lounge to check out our sponsors and exhibitors, or network with other attendees. Join the conversation on Twitter with hashtag CENSIM15.